Okay, it is my pleasure to introduce Paul Wilson, our speaker tonight. Paul grew up here in Humboldt County. He received a BS degree in botany from Humboldt State, and then he went on to graduate school at Stony Brook University in Long Island. And then for years, he worked as a professor of ecology and evolution at Cal State Northridge. His research originally focused on plant evolution, which is what we're going to be hearing about tonight. And then he gradually transitioned into research on bryophytes, and he was the president of the CNPS Bryophyte chapter for several years. So Paul has now retired. He's very happy to be back here in Humboldt County, and we're very happy to have him because he has stepped forward to take my place as program chair starting next fall. So he'll be our program chair and publicity coordinator. So please join me in welcoming Paul Wilson.
Hey, Zoomers, can you hear me?
I can hear you.
All right. I'm going to turn this on. Oh, great. Somebody took the lights. Ooh, so I have control. Cool. Okay, so I'm going to be speaking about convergent evolution in this group of flowers that I'll call penstemons. It includes the genus Penstemon as well as some other genera that are closely related. And in this group, well, to preview, it started out bee pollinated, and then there's been over and over again the evolution of hummingbird pollination. There's a lot of species. They all live in North America, and it's thought that this radiation is pretty recent. I mean, as radiations go. Okay, so let's see if I can control this. If I go like this, oh yeah. So I'll be talking about the research of a whole bunch of people. James Thompson and Maria Clara Castellanos and I worked on this for about a decade in the late 90s and early aughts. And we were studying kind of like sexual selection in the flowers. And then Andrea Wolf was involved in developing the evolutionary tree and doing other sort of things with DNA. And then we kind of got out of the business, and Mark Rauscher and Carrie Wissinger took over. And really, this is a talk that could be viewed as a review of Carrie's research. She's done tremendous things. So moving on, I can use that one too. Okay, so there's about 300 species of penstemons and related genera in North America. Most are bee or wasp pollinated. There's at least 30 that are more or less hummingbird pollinated. And some of them are, I guess you would say, they've taken on hummingbirds, but they haven't given up bees. And then a bunch of them have more or less given up bees. So this one here is one I would say is specialized on hummingbirds. And this one is specialized on bees and wasps. And let's just jump into it. So one of the first things that we did is went around and collected lots and lots of species and got their DNA. And then Andy developed this evolutionary tree from DNA sequences. So this tree here is based on a nuclear region that's probably more or less neutral. And you can see that hummingbird pollination arises here, there, and yonder, apparently separate originations. Maybe I can do better with this one. See, like this little group is separate from that group. And then we could also get DNA sequence from the chloroplast and could build another tree. Now, these don't have the same representation of species, and neither tree has all the species. So, you know, it's a little bit partial, but we did the best we could at the time. So now if we take these two evolutionary trees and put them together, then we can try to figure out the number of times you've had the origination of hummingbird pollination. We're trying to, like get each one and how much evidence there is for it. And there's a whole bunch of statistics that we could do for that. The best estimate based on these data, which is not all of the species, is 21 separate originations. And we feel like there's overwhelming evidence for 10 of them. So like 10 that I wouldn't say you could contest. One of the ways that we do this is to take two species, say Centranthifolius and Rostriflorus there, and then hold them together so that they have to be related to one another, and then try to figure out how much longer would the evolutionary tree have to be if we did that. And it would be the nuclear tree would be 6 steps longer, and the chloroplast tree would also be 6 steps longer. So that's like 12 extra evolutionary changes you would have to invoke in order to bring together Rostriflorus and Centranthifolius. And then you do that over and over and over again for a whole bunch of different combinations to try to figure out which ones, are very convincing versus ones that are just the best guess. And that's how we kind of arrive at these 10 that I think are beyond doubt. Okay, so that was sort of the technology at the turn of the millennium. But then Carrie and Mark have much better technology. They haven't sampled the same species. So this is a tree based on their data. And they have data from, oh boy, no. That's the wrong button, definitely. Okay, let's try this. They have data throughout the genome, a bunch of little different genes all over, like they've got hundreds and hundreds of them, probably thousands. And this is just the core group of penstemons. So it's not all the ones that are farther out. If you had all the ones that were farther out, you would get a whole bunch more originations of hummingbird pollination. And, maybe they'll do that sometime. Maybe it's not that exciting, but it would kind of elevate the whole story to maybe a kind of textbook level. Yeah, go ahead, Dave.
Is it known what genes responsible for change?
Oh yeah, we'll get to it. Yeah, we don't know the names of the genes, but we know very, very small regions that they're from. Yeah. And in the case of color, we do know. So we'll get to that. Okay, so if you take Carrie's data, then you can do this estimation where you try to figure out the rate of transition from bee pollination to hummingbird pollination, that's the green arrow, compared to the rate of transition from hummingbird pollination to bee pollination, that's the pink arrow. Now, we actually don't have any evidence that there ever is a transition from hummingbird to bee pollination. But it's very hard statistically to tell a reversal from a convergence. Like they both require the same number of steps usually. Anyway, like our best estimate is that there's 0 reversals. And that's true throughout the whole data set, not just the sub data set that they're able to do this analysis on. We also get out the rate of speciation. I don't like this thing. I think I'm going to stop using it. We also get out the rate of speciation and the rate of extinction. And the difference between those is the diversification rate. So sometimes it's hard to tell speciation from the opposite of extinction. But you do get the difference, and that's diversification. So let's move out to results. You take all Carrie's data, and she puts it through this thing and looks at a whole bunch of outcomes, and it kind of boils down to a couple of graphs. So here's one of them comparing the transitions from bee pollination to hummingbird pollination, that's in green. to the transitions from hummingbird pollination to bee pollination. That's in this pinkish color. And seems like there's a difference. It's not statistically significant, which is a little bit worrisome. And this would be a reason to go look at all of those other species that are beyond the core group. And then here's the exciting new finding, and this is statistically significant, and that is that hummingbird pollination seems to have a lower diversification rate than bee pollination, and this is probably because of the speciation rate. So in other words, over and over again, you have the evolution of hummingbird pollination, and then those lineages that have become to be pollinated by hummingbirds, they then stop making species. I mean, I'm exaggerating a little bit. Whereas the lineages that are bee pollinated are just cranking out new species all the time, have a very high speciation rate. So that's kind of cool. And yeah, well, we'll move on. Okay, so this is what I've told you so far, that penstemons, bee to bird pollination could have occurred as many as 20 times, and there's overwhelming evidence for 10 of them. Bird-adapted flowers have this dampened diversification rate, probably due to a dampened speciation rate. It's hard to distinguish convergence from reversal, but there's no evidence for any reversal. That is, there's no case where you can point to and say, oh, it's likely that we had a hummingbird pollinated plant, and then out of that arose a bee pollinated plant. It always seems to go the other direction. All right, moving on to the next little section. So once we have this evolutionary tree, you can go to parts of it and pick out a lineage that's hummingbird pollinated, and then a closely related lineage that is bee pollinated. And that I would call an evolutionary block, or this is sort of like block sampling. So here's an example, and then here's another example, and so on. So on this slide, I've got 7 examples where you've got closely related flowers, where one is bee pollinated, one is hummingbird pollinated. And this allows us to delve into the many characters that are involved in the pollination syndrome and see how they've, I guess, co-adapted to one another, or both, or co-adapted to their pollinators, and therefore come to be in a syndrome with one another. Okay, so I break this down to what I call systematic rules. And the first one here is that bird flowers tend to be, if anything, more red or orange than bee pollinated relatives. People have suggested this for, time out of mind, and we're able to go into our data set and say that indeed that's the case. But now I'll go through a whole bunch of other characters. So the next set of characters to think about is nectar. And people have suggested that bird flowers tend to produce more nectar that's more dilute, and it's relatively sucrose rich. So if we look at these graphs, the one on the driver's side here shows that the hummingbird pollinated flowers produce more nectar than the bee pollinated flowers. And then the one in the center shows that the hummingbird pollinated flowers have a higher concentration of nectar than, the bee pollinated flowers have a higher concentration of nectar than the hummingbird flowers. So the hummingbird flowers, it's like 25%, which is sweet, but not so sweet to be called syrup. Whereas at least some of the bee pollinated plants make nectar that it would be a bit hard to lap it up if you were you. But evidently bees are pretty good at lapping it up. And then there's also a difference in the relative amount of sucrose versus hexose, and hummingbirds seem to like they're energetically equivalent, but the hummingbirds seem to like the nectar that is rich in sucrose, which is a disaccharide, whereas the bees seem to like maybe fructose or some other hexose. So you can see how these systematic rules work. You just kind of go through each character that people have made a hypothesis about and then check it against, the 10 cases and see if it works out the way people have suggested. And it turns out that it pretty much always does. So here's some more characters. So bird flowers tend to have narrower tubes, whereas the bee flowers in Penstemon, the bee like crawls into the tube and it's more like a vestibule. The bird flowers tend to have more exerted anthers and stigmas. So if you look at this bird flower, see the anthers here and the stigma here are sticking way out. And then the next couple of ones, you have to put some oars in there or something like that. But the typical bee pollinated Penstemon has this great big landing platform that the bee, comes down on and then kind of wriggles into the flower by manhandling. Well, I guess woman handling if you're a bee. The male bees don't do really anything. Yeah, that's another story. Whereas the hummingbird pollinated flowers, there's lots of different things that the lower lip turns into, but very generally, it's no longer a landing platform. So sometimes the lip folds all the way back. Sometimes it's just reduced to a few little lobes, etc. And then the next character to consider is that the hummingbird flowers tend to be more inclined, like they might be like this, or they might be floppy, whereas the bee pollinated flowers are held rigidly horizontal, maybe just a little bit inclined. And well, anyway, that turns out well. And then James had this, James had this whole idea about how we could use Penstemon to test what amounts to sexual selection theory in plants. And that turns out to be true too. I'll give you the short version. Bird flowers tend to have anthers that open wider or more simultaneously than bee pollinated relatives. And then now I'll show you some pictures of it. So just while I'm showing the pictures, think about it this way. Antlers in deer are these structures that were shaped by male deer fighting with one another, competing with one another, either actually or ritualistically or whatever. So they're the product of sexual selection. And James, and the rest of us now, I guess, too, think that anthers are similarly the product of flowers competing with one another in terms of male function, in terms of like getting their pollen grains out to all the stigmas in the world. And that's why Penstemon has these ornate anthers. So that's kind of the general idea. And then now let's look at some Penstemon anthers. So here's a pair, one of these blocks that we're using to test the systematic rules. And the lower one, Penstemon kunthii, the anthers, when they're open, they look like tostadas. The pollen's just, on this flat thing. Whereas Penstemon gentrii, which is a very close relative, it looks like a series of tacos where the pollen is in a bit of a crevice. So that one matches James's hypothesis. And then here's another one that also matches James's hypothesis. Remember that Pinifolius and kunthii are not close relatives. They both arose from bee pollination, and they are respectively close relatives to the bee pollinated things that are above them. But again, you sort of see, oh, the Pinifolius, it opens more widely. And then those are kind of boring anthers, as anthers go in Penstemon. But these ones are, just ultimately cool. So Rostra floris here, it doesn't open widely, but it opens a lot more widely than Penstemon ledus, which just cracks open a little bit and pushes a little teeny bit of pollen out on each bee visit, dosing them over and over and over again, like hundreds of bee visits could have pollen removed. If a bee came up to Rostra floris and started removing pollen, it wouldn't, it would get most of it in the first visit. But a hummingbird comes up to Rostra floris, and hummingbirds are not at all interested in pollen. They're trying to get their get nectar there. And so this anther brushes against their feathers on their forehead, and the pollen is removed that way. And we think that it takes a while to exhaust the pollen supply in Penstemon Rostra floris, even though it opens much more widely than Penstemon ledus. And then these other two we call origami anthers. And Penstemon lancia, let's see if I can move this. All right, never mind. The lower one opens up more widely than the upper one. Anyway, I'm getting boring. So here's a bunch of them, right? And if we look at 18 pairs, then we find that there's a statistically significant pattern to this systematic rule. There are a couple no's, and then there's several ties. And at least with one of these ties, we had a student, and she just went out and camped next to the two penstemons and looked at them every 20 minutes, like pretty much day and night. And she found that in the case where the anthers opened up to the same degree, the bird pollinated species kind of staggered it. So there's 4 anthers, and what would happen, oh no, not the bird, the bee pollinated species. So it started out, the bee pollinated species staggered the anther presentation. So it would open up like one anther, and then 8 hours later it would open up another anther, and then the next morning it would open up another anther, and then the next afternoon it would open up another anther. So there was gradual staggered presentation of pollen by the bee pollinated species. And then the closely related bird pollinated species, I think she never found an anther that was partially open. They pretty much opened up their anthers really quickly and very close to at the same time. The front 2 would open first, and then the latter 2 would open an hour later or something like that. So I'd count that as a yes, even though it was a tie, if you just looked at it structurally and not over time. Anyway, it made the paper longer. Okay, so we've tested our idea of sexual selection. And if you ever want me to just talk about sexual selection, I could come back and give a talk like that, but it probably wouldn't have very many pictures of flowers. Okay, let's move on to pigment. So Dave was asking about the genes involved and whatever. So the pigment that's responsible for the color is an anthocyanin, and here's the whole anthocyanin enzymatic pathway. But it turns out most of this you don't need to know. So here's a simplified version. In particular, if this enzyme F3'5' is working, then you end up with something that is the color of a larkspur. And if it's not working, then you end up with something that's the color of a geranium. And this has been known for a long time in general. But what Mark and Carrie did is they went to all of these penstemons and they sequenced these genes, a bunch of these genes that are responsible for these enzymes. And here's what they found. They found that it was always this F3'5' gene that was the one that was responsible for the change. Various things happen, like sometimes you get a premature stop codon, and that, of course, makes the enzyme not work because you don't get the whole gene expressed. And then sometimes you have a frame shift, and that just makes it so that everything after that point is nonsense and doesn't make an enzyme that works, and so on. So a lot of these are well on their way to becoming pseudogenes. But then there's a few that maybe not, like these top 2, maybe not. So, Carrie went back and spent huge numbers of hours in the lab doing enzyme assays. And she found that all except the top one had no enzyme function that was detectable. And then the top one had partial enzyme function, but less than the bee adapted relative. So they're always compared to some bee adapted relative or other. So anyway, it's kind of cool. It's like this one gene is responsible for the change in color, and it's pretty easy to have a mutation that changes the color. And it's pretty hard to imagine it turning around and going the other direction. So here it is in words. She asked the question, why are inactivating mutations to F, 3', 5' H so predictable? And here's some reasons. First, it's a pretty simple genetic pathway, like it's just like one locus. You don't have to worry about all these other loci that might compensate or something. It's a huge mutational target, like anywhere in this gene. If you mess it up, the enzyme's gone. And then there's few deleterious correlated pleiotropic effects. So it's not like when they shift over from being violet to being red, they start getting sunburned or something. They still have anthocyanins that just doesn't get shunted into that blue color. So we conclude that these features facilitate transitions from blue to red, but hinder reversals in color. And so that's sort of a partial explanation for why we seem to have great lability moving towards hummingbird pollination, and yet great constraint getting out of hummingbird pollination. Because there's other external reasons too, and that is maybe just hummingbirds are great pollinators. Like, why would you ever evolve away from that? But there's this internal reason as well. Okay, so adding to my little list of take-home messages, we have the genetics of flower color provides this explanation for pollinator shifts without reversals. And now let's go into, like, so far I haven't actually told you anything about, like studying visitation or pollination. And we have a lot of data on that, but I thought I should tell you at least a little bit about it. So here's a hummingbird visiting a flower, and yeah, here's bees visiting flowers. The Pseudomyceras is a wasp, and it's not closely related to bees, but it kind of has become bee-like. So it feeds its offspring pollen just like bees feed their offspring pollen, and it forages actively on flowers. And this one is more or less a specialist on penstemon. And then bumblebees, everybody knows bumblebees. Anthopteras also visit penstemons, and they're about the same size as a bumblebee. And then there's lots of osmias, which are medium-sized bees that love penstemons. A whole bunch of species, which kind of drove us crazy for a while. But you don't have to know them. I mean, it's good to know them, especially if you like bees. But from a penstemon's point of view, they're probably all the same. And then there's loads of pollen collector bees that are like little sweat bees and stuff like that. Kind of a heterogeneous group, but we think that they're like, most cases, they're kind of evil, ugly bees. They're in there pigging out on pollen and not moving very much of it to stigmas, and they're just like parasites on your genitalia if you're a flower. Okay, so oh yeah, what do we do with this? So it was pretty easy. If you know pollinators, and you just stand at a patch of flowers for 30 minutes and you write down everything that comes to them and what they're doing. And we did this for 84 species, hundreds of visitor censuses. And that's sort of the basis of our knowledge. And more could be done on this too. Like there's still some more penstemons out there that we've never been able to get this done to. Okay, here's sort of a subgroup of that. This is 49 species. And we measured the 9 floral characters that I told you about before that have to do with the syndrome. And then you put them on this graph. Don't worry about what the axes mean, but basically you get a group of hummingbird pollinated species, and then a big gap in the middle, and then a group of bee pollinated species. That's based only on the floral characters. And then here's the same graph with the visitation on top of it. So that vector that goes out more or less parallel to axis 1 is hummingbird pollination. If you're high on the high value of axis 1, you get lots and lots of hummingbird visits and not very many bee visits. If you're low on axis 1, you get lots of bee visits and not very many hummingbird visits. Then axis 2 has it's kind of the difference between a great big bees, xylocopa or like carpenter bees, versus osmias, which are medium-sized bees. And yeah, it works. The visitors are not as distinct as the flowers, but anyway, we weren't just assuming things that we weren't just assuming that the animals do what we think they do. We did actually have a lot of data to base that on. Now, that was sort of a subset. I think, what did I say, 49 species or something. And it did include all of these species, but all of these species, well, these ones that I'm showing here, show some ones that I would say are kind of in between. So like I showed you that there's this big gap, but I really still want to temper it with saying that there is some in between in this. It's like Hartwigii here. You might have this in your garden. Hartwigii and ginginoides have great big flowers. They naturally grow near Mexico City in the great big mountains there. And we went down there and studied them in the wild. But people love to plant these in their gardens. So there's all sorts of hybrids of these things also. And I would say Hartwigii is sort of halfway to hummingbird pollination. Hummingbirds love it. Bees can still get inside of it. The close relative fasciculatus, that's a totally hummingbird pollinated thing. You know, only a small bee could get into, only a very small bee could get down to the nectar. And then Kunthii is another kind of intermediate, you know, like floral morphology. It has exerted anthers and all that stuff, and it's red, but a bee could still get inside of it. And so I think there's this process, which is sort of the two-step of it, that you start out being bee pollinated. First, you have despecialization, where the flower adapts to hummingbirds. And then at that point, it's attracting both hummingbirds and bees. And then sometimes you have respecialization, where we think the bees become parasites in terms of sexual selection, and the flowers adapt or specialize on hummingbird pollination. So to boil it down, the syndromes are more or less distinct. They're definitely different. And visitation is a little bit less so. Like you do have hummingbirds, of course, that visit bee-pollinated penstemons. And sometimes you have bees that visit hummingbird-pollinated penstemons. Okay, moving on. Carrie had a student who was able to like digitize all of the herbarium records of loads and loads of penstemons and figure out what the median elevation and the median latitude was for each of a bunch of species. And, you know, maybe not too surprisingly, the hummingbird pollinated ones tend to have lower elevations and be at lower latitudes. And you might know that there's lots and lots of hummingbirds that you can find in this region, and, you know, not so many hummingbirds in Alaska. And so, okay, that was kind of interesting. But then they dissected it a bit more. They kind of like titrated this into different parts. The first thing that they tried was just like what we usually do. You get a hummingbird pollinated species and a bee pollinated species, and you ask if they have different elevations. And that doesn't work. So here's those data. Hummingbird and bee pollinated species, you know, kind of paired up in the usual way, block way. That doesn't work. So then they looked at different bee pollinated species, ones that were closely related, that is sister lineages to hummingbird pollinated species. That's the one that's kind of in violet here. So a bee pollinated species that's related to a hummingbird pollinated species versus penstemons that are not particularly related to hummingbird pollinated species. And there you find the difference. So it's like what happens is the lineages, in order to evolve hummingbird pollination, they have to first, as a precursor, evolve to live at lower elevations and lower latitudes. And then sometimes hummingbird pollination arises, but it doesn't arise at high latitudes and high elevations. So here's like the little scenario. It goes from maybe the northern Sierra Nevada down to the Sky Islands of Arizona, and then it splits into hummingbird and bee pollinated syndromes. So expanding to low latitude and elevation is a precursor to evolving bird flowers. It's not part of the syndrome. All right, moving on. Yeah, I don't know. You might feel a little bit tired at this point, and I put the worst part, the hardest thing at the end, but I didn't want to like lose you by putting it at the beginning. And so, just bear with me. I'll get to the end at some point. So Carrie got really interested or really able to study this group that has some very, very close relatives, Penstemon barbatus, and then a couple of bee pollinated species that are very close relatives. And this isn't really an evolutionary tree, but you can see that Penstemon barbatus is sometimes related to Penstemon virgatus, and then sometimes it's related to other Penstemon virgatus. Like it's like two different parts in different places, Arizona versus along the Continental Divide. Yeah, this is based on the same, like thousands of SNPs. Yeah. Anyway, the story will develop. You'll see it's kind of cool. But anyway, it means that the things that we call species or syndromes or whatever, it's all kind of mushed together at that point. It's maintained in the face of some kind of relatedness that is counterintuitive. And that could be gene flow. It's like the syndrome is maintained in the face of gene flow, or it could be that the polymorphism the evolution is so recent that the polymorphism kind of persists through the syndrome formation. But either way, either way, the genetics is not doing the same thing as the pollination syndromes, even though these flowers are like, no way, under any circumstance, are we putting these in the same species. Like, even though there's a few characters that, there's only a few characters that define the syndrome, and if you didn't have those, you wouldn't really know what to do with them other than put them in the same species. Okay, so, here we go. So like they have thousands of genetic loci, and each one of these things is, let's just call it like a chromosome. It's a linkage group, and there's 8 of these linkage groups, and they tend to show more recombination on the ends than in the middle. Each little teeny dot, each little teeny blue dot is a genetic locus, and the vast majority of the genetic loci are neutral. They never make any enzyme or anything. And so what she's done is figured out for each genetic locus, how does it correlate with the syndrome or the species? And if it's high on the graph, like a little point, this little point here, then that means that it's a species indicative, or yeah, it's a genetic marker you could use to identify the species. And if it's any of this big cloud of blue dots, it's not a genetic marker that you could use to identify the species. And then the lower part of the diagram is just, expanding greatly on something. So like this part here is just taking this little bit here and blowing it up so that we can look at it. And in this region here, that's where the flower color gene is. And there's one of these species distinctive genetic loci that is super correlated with flower color. Like, we don't know if it's actually the same gene or not, but it's right in the same place. And there's two of them that are right next to nectar volume. And then there's one of them that's associated with flower width. Those three characters are all single genetic loci Mendelian type of things. Then the other ones, there's a bunch of other ones where it's not a single genetic locus, but she kind of knows, like, oh, there's these 2 or 3 places on the chromosome that explain quite a bit of how the hybrids segregate. And so I don't know, 21 is like the magic number of the evening, but there's 21 of these genetic loci that differentiate the species or the syndrome or whatever it is. And almost all of them are accounted for by the syndrome traits that she's able to measure. So it's like, there's no other genetic loci other than the syndrome traits by which you could tell these species apart. After looking at thousands of them. Okay, so only 21 SNPs are diagnostic of species. Most are tightly linked to genes attributable to syndrome characters. The pollinator shift seems very close to the cutting edge of speciation, perhaps even being, that is, the species being recreated generation to generation despite gene flow. This is nothing I would have argued for. Like, I had to be dragged kicking and screaming to believe this data, because I think, no. Speciation happens, and then sometimes flowers shift over to hummingbird pollination. But in this case, I can't really see it that way. Like, it's a very strong syndrome distinction, and there's nothing else in there that you would call speciation in the sense of something responsible for this diagnosis of the species. I mean, of course, there might be something buried deep down that has to do, like some genetic locus we haven't found, that has to do with why, one kind has to live in certain sandy soil and one kind has to live in clayey soil that we haven't found yet. But even if so, it's just going to be another one of these. It's just going to be like one more locus. It's not going to be the whole genome. The weight of the genome, the weight of all of the genetics says that this is continuous variation. So anyway, not what I thought was going to happen. So in the case of Barbados, the pollinator shift is very close to speciation. Like, the syndrome and the species are pretty similar. And you could, maybe I shouldn't use the word speciation here, but all I mean is just the thing that's responsible for us calling them different species. All right. So there's this slide of all the things you should know that will be on the test. And then here it is in white. And I'll put this up, but I think I'm going to just sort of anticipate a question rather than let you ask it. And that is like, oh, what about all the other pollinators? So what about moths? There is a penstemon that's white. And so somebody saw it. There's a bunch of them that are white, but this one's white and a little bit narrow. And so somebody said, oh, it must be moth pollinated. But then people went out and like spent years watching the pollinators, and no, it's not moth pollinated. Like, occasionally you have a moth coming to it. Most of the pollination is by bees. And then, so that one you can pretty much just ignore. But then this is a pair of 2 species that are really cool, interesting species. They branch off pretty early in the core group. They're visited by, they pretty much given up attracting things based on nectar. They basically don't make nectar as far as I can tell. Either that or it was a drought year when I was studying it. But anyway, there's no nectar there. And they are still visited, and they're visited by pollen collecting bees, the same pollen collecting bees that visit a lot of other things that we think of as parasites. Maybe they're not parasites here. In this case, maybe they're the real pollinators of these two species. And then they're also pollinated by these tiny little bee flies that go into these flowers to sleep and mate. And I don't know much about them, but somebody claimed that they were the pollinators of these things, and they certainly do visit them. So I think that they're probably pollinated by, both of those things. And that's the only thing that I would, kind of count as not part of the dichotomy between hummingbirds and bees and the despecialized ones in between. So anyway, I think that there's a lot of lability here. You got a lot of opportunity for evolution to happen, but it's very constrained. Like it never reverses as far as we can tell, and it never goes off and becomes bat pollination or anything like that. And that is all I have to say about that.
Well,
Yeah, it's a pretty cool story. I'm glad that Carrie took it over. Yeah, Ken?
you've been able to tease out the chronology here. Have you been able to? Is this working?
I don't hear it.
Have you been able to tease out the chronology of this at all? Specifically, how long ago did these shifts occur? Were they more or less synchronous? And how closely do they align with the appearance of hummingbirds?
Oh, okay. I don't think we can tell at the level that about synchronicity. I mean, that's like too fine a scale, but we believe that all of this is happening, like way after there's hummingbirds all over the place. Yeah. I think, and also, I also think that at least in terms of functional groups, the bees predate this. And so it's not like there's coevolution between the pollinators and the flowers, except for maybe that wasp. Yeah, it's more like the pollinator scene was set and then the flowers diversified. Well, it wasn't just the last ice age, but it doesn't go back tens of millions of years. So, yeah. Yeah, Donna?
So early on in the talk, you mentioned that, if I remember correctly, that bird-pollinated penstemons don't seem to speciate as frequently as bee-pollinated.
Yeah, which I think is a really cool result. Mm-hmm.
Yeah, do you have
Yeah, so the idea, like I don't know how to test this or anything, but the idea is just that birds are responsible for much greater gene flow. They can just like move pollen around between patches easily, and that somehow inhibits the formation of new bird pollinated species. Also, the birds in the US anyway are kind of all the same. I mean, they're different colors and stuff. But and it turns out that when people try to do this type of thing in Central America and tropical South America, the rules don't work. And that could be because hummingbirds are really diverse group. They're not, they're not all just the same size, migratory, blah, blah, blah. You get some really different types of hummingbirds in Costa Rica, Ecuador.
The other question I had was, so are there other species of flowering plants where you have this kind of alternation between bird pollination and bee pollination?
Yeah, so the bifurcation of the niche is the way I would think of it. It's not just penstemons. There's loads and loads of other groups where you'll have, a case anyway, of that. Maybe Aquilegia would be a good case. Delphinium, our local delphinium, Delphinium nudicale versus standard delphinium. And then in Southern California is Delphinium cardinale, which looks like nudicale. And yeah, back when I was editor of Darlingtonia, I wrote a little like catalog of all of these things locally that you could see hummingbird pollination versus something. There's also a lot of ones that have to do with hawk moths, and we don't get hawk moths on Penstemon. So yeah, like the Colorado columbine, that's adapted to hawk moths. It's also visited by bees who do a lot of pollination, but most of the characters have to do with hawk moth pollination, whereas Aquilegia formosa, that's hummingbird pollinated.
Okay, thank you.
Is there anybody, do we have anything on Zoom land? Is somebody actually reading the comments or something? What's happening?
Paul,
Yeah.
I don't see any comments in the chat yet on Zoom land.
Okay, yeah, Jillian?
Well, I was interested in.
You mean what being it parasitic on the flower? Yeah, okay, so if you're in a situation where there's reliably high visitation by hummingbirds and there's bees that are present, and the bees are like osmia bees. Well, osmias are these little solitary bees. They're not tiny, but they're, smaller than a honeybee. And what's the job of an osmia? The job of a female osmia is to collect pollen. They also drink nectar to keep themselves going, but they collect pollen in order to take home to build baby bee bodies. And so they lay an egg, and then they make a pollen wad and put it on that egg. They sort of seal that one up, then they lay another egg, put a great big pollen wad on it, seal, so they are covered with hairs, and their behavior is to go up to a flower, kind of go like this. The pollen gets all over their body, then they back up and they groom it a lot, and they get it, kind of contained. So they're removing pollen from the system heavily, whereas a hummingbird doesn't care about pollen, like it eats insects. That's how it gets its protein. They're just like, I want cookies. Give me more, more sugar now. And so they get pollen on their foreheads, and it has a tendency to just keep on going. We only measured it out to 18 more visits, but we got tired of doing this. But we think that if you extrapolate the curve out, it probably goes out to dozens and dozens of subsequent visits. That pollen from one flower could end up on the 50th flower, the 50th plant down the line. And so they're not wasteful. So if you have enough hummingbirds, it's going to make the bees be kind of parasites from the point of view of sexual selection and male function. Yeah.
And thinking about have changed the ventilation.
You mean like methylation as in deactivating? Yeah. Not that I know of. That would be a question for Mark, I think. Yeah. Like, yeah, I pretty quickly get out of my depth. But he might know, but I don't know. Yeah. Behind you, I think.
I'm just curious if seed dispersal is different between bee and hummingbird pollinated species, and if that could have anything to do with differences in speciation rates, especially like allopatric speciation.
Right. So we weighed the seeds and counted there's little penstemons and big penstemons. I mean, yeah, it's like, I don't think so. But if you think about it in terms of the elevation and latitude bit, it might have something to do with a precursor. Like maybe only the ones that make big seeds have the potential to shift over. But then the sister groups, I don't think so. Yeah. There are all these little penstemons. You might know them. Like if you go up the Cascades, there's all these little teeny penstemons that, okay, a hummingbird might check them out, but it probably wouldn't, you wouldn't say they were adapted to hummingbirds. That's not the penstemons we're talking about. We're talking about ones that have nice big flowers. Like Penstemon procerus, people in the room know Penstemon procerus or Rydbergii, one of those two. They're like, step on them. Anyone else? No? Yeah, Dr. Waters. Oh, yeah. Yeah. I can send you, just send me an email. I'll send you a reading list. Yeah. Yeah, and I have to reread them too, like I even if the papers I wrote, I have to reread. I had to reread them in order to make this talk. So yeah, yeah. They're dense. Yeah, yeah. And there's also a bunch on the sexual selection thing. But if you get into that, then that's a whole nother can of pollen. Maybe pollen tubes. Yeah. Okay, well, let's put up the chairs.